We've got here a bit of a big picture view of proteins, so now we're going to go down real small and look at the amino acids that make up these proteins. So I've got a few here, and we're going to look now at each individual atom and talk a little bit more about these amino acids. So first of all, there's some things you know about amino acids already. Amino should remind you of amine functional groups, and acid should remind you of carboxylic functional group or carboxylic acid functional groups. And sure enough, we're going to see that we have amines and carboxylic acids in these amino acids. So first, I'm just going to draw you a generic amino acid. So this would work for any amino acid. In that generic amino acid, I'm going to have that amine group, so NH2, written backwards, H2N, and then a carbon, and that carbon is connected to a carboxyl group, and then a hydrogen, and then an R. So this would be a generic amino acid here. We have our amine group, we have our carboxylic acid group, we have our R group, which will be the rest of the molecule. That was kind of our filler that we've seen before. And if you notice here, as long as the R is not a hydrogen, this carbon is generally going to be a chiral center. And you'll see sometimes written for all of our amino acids names that they have an L in front of them. So the biologically active amino acids, or the functional ones, are L-alanine and L-glycine and L-aspartic acid. And that is tied to this chiral center here, the orientation around it. So that's our chiral center. We also call that an alpha carbon. If you remember, the omega carbons were really far from carboxylic acids in chapter 15. And so at the other end, the carbon that's right next to a carboxylic acid is called the alpha carbon. So these are sometimes called alpha amino acids as well. This R group, then, we want to focus on a little bit. This is what we call our side chain. And once we string these amino acids into a long polymer, right, a protein is just going to be a long chain of amino acids. The backbone here on top is going to get tied up in that backbone here, and what you'll have is these free side chains. And so the side chains are where we really want to put our focus to describe their polarity. Because if those side chains are polar, like we talked about on the last slide, those polar side chains are going to fold up on the outside of a globular protein, and the nonpolar or hydrophobic amino acid side chains will fold up on the inside of the protein. So we want to be able to identify, is a side chain polar or not? And this is just going to be recycling everything you learned in chapters 3 and 4 and 7 about polar and nonpolar. So the first piece that you want to look at as we look at these 4 amino acids is find the side chain. So, oh, and actually before we do that, one more thing here. You'll notice the generic amino acid I drew doesn't have charges, and then all of these have charges on them. This is actually how we would find them in nature. So acids like to donate protons to bases, right? So this H gets donated over here. It's an acid-base reaction all within the same molecule. And so what I see here is I see charges which should impact the water solubility too, right? What's a charge going to do to the water solubility? It's going to make it more polar, right? So all of these where I have both ions are called Zwitter ions. Comes from German, I think. And the Zwitter ions have both a positive and a negative charge in them because that proton has moved from the acid over to the base. Now, we use the words acid and base kind of loosely when we're talking about amino acids. Even though this has lost its H and is technically the conjugate base, we still call it an acid. And even though this is protonated, we still call it the base. So don't get thrown by that. It's a little bit different than what we've done so far. So as I look at this, I want to find the side chains. So the non-backbone piece. So here's the backbone, here's the side chain. Here's the backbone, here's the side chain. Here's the backbone, here's the side chain. And we want to be able to describe those side chains, again, as polar or nonpolar, and also if they're acidic or basic. So once we can do that, then we can go to your whole huge page of amino acids and classify every single side chain. And that will help us, again, know where these are going to fold up to. So if I look just at what's in this green circle here, I would say that this is nonpolar. It's all C's and H's, right? And C's and H's we know are nonpolar. So we have some side chains that are nonpolar. If I look at this one here, this COO minus should jump out at you as, hey, that's an acid, right? An acid actually in my side chain. And I have a lot of polar bonds here, C to O, in 2 directions. So I would say that this is a polar side chain, just within the green here. And even though it's deprotonated, I still call it polar acidic, all right? So there's that acid that we've seen before. Down here, I can see that there's a nitrogen and it's been protonated. So that tells me, it's a good proton acceptor. Also, I see that NH is a very polar bond. So if I can see a protonated amine, this is going to be polar basic. So a polar side chain, but basic. And then last of all, down here, I noticed that I have a carbonyl, I have a nitrogen. This is an amide actually in the side chain. Definitely a bunch of polar bonds. And this is where you need to remember something you learned back in chapter 13, that amides are not basic. So they tempt you, but they are not basic, even though they have a nitrogen. Anything that's a base that we see in this chapter that is a polar basic side chain will have a protonated amine in it. So those are our 4 categories for our side chains. So your task now is to go through your 20 amino acids. This is on page 73 in your workbook. It's also a part of your lecture slides here. And go through for every amino acid on this page and find those side chains. So you're going to do something like this for every amino acid on here and classify them as polar or non-polar. And I'm just going to look at a couple with you here real quickly, and the rest I will leave for you to do. Methionine here, these are in alphabetical order, and this is a sheet that you want to have handy with you every time you take a quiz or a test. So methionine has sulfurs and carbons. If I look up the electronegativity differences between sulfur and carbon, that was something we did back in chapter 3 with polar bonds. I would find that those bonds are sharing electrons very evenly. So I would say that this is a non-polar side chain. And then there's a couple more that I want to look at down here. Tryptophan, we talk about around Thanksgiving sometimes. So side chain here, and here's a tyrosine. If you notice, each of these has a polar bond, NH or OH, but it has maybe 7 or more carbons with it. And so if you remember back to alcohols, if you're more than 4, this OH kind of loses its power. So I would say that these side chains are both majority non-polar. However, if you put them near something polar, a side chain that's polar, we would notice that this tiny bit of polar here would gladly go hydrogen bond with, say, an OH on serine. So those are just a couple of the strange ones to look for. Otherwise, you're going to classify everything on this page as either non-polar, polar, polar acidic, or polar basic. So find all those side chains, circle them, and tell me, okay, that's all C's and H's, so that's non-polar. And then bring any questions that you have to class.